Ok, puoi partire.
Okay, so just to introduce this colloquium with a few words. So I think that this colloquium could be viewed as a partial answer to the following question: how to find technological application of quantum mechanics exploiting its most counterintuitive aspects, such as the appearance of a non-classical degrees of freedom, the spin, the superposition of states, and the entanglement. The theme is presently worldwide at the center of a big effort, also on the financial side. Besides quantum computer and quantum cryptography, another key actor in this effort is quantum sensing. The use of quantum systems as sensors for various physical quantities, going beyond the classical performance, taking advantage of the strong quantum sensitivity to external disturbances, typically due to discrete features appearing in quantum physics and reaching the scale of manipulation of individual atoms. It is a great pleasure and honor to have here as a speaker of this colloquium, Fedor Jelezko, which is a world-recognized leading expert in this branch of quantum technology. He studied in Minsk, got his PhD at Bordeaux, later becoming professor in Stuttgart, and presently he is a director of the Institute of Quantum and Optics at the University of Ulm. For his achievement in solid-state quantum optics and quantum technology, he received several prizes. Let me just mention the Walter Schott Prize of the German Physical Society and the Spolsky-Reban Personov Medal. His research interests are at the interface of fundamental quantum physics and applications of quantum technologies to quantum sensing, information processing, and communication, and even to life science. Today, he will introduce us to the fascinating world of application of spin qubits. So please, Professor Jelezko.
Thank you very much for this kind introduction, for invitation to give this seminar. It's a pleasure for me. I apologize for not being able to come this time owing to travel restrictions, but I would love to come to Padova and learn more about your research. Today, I'm going to introduce a diamond as a platform for quantum technology, how to control single atoms in diamond and how to make use of it, of these control tools, for reaching new regimes in the field of computing and in the field of sensing. And I will start to share the screen now, and the first slide show you one experiment in our lab where we were using natural crystals to explore a new defect that we are not able to create yet artificially. And this crystal will be placed now in a sample holder, where you see there is a microwave line to control individual atoms in this diamond. But to motivate this work, I would like to put it in the context of solid-state quantum technology. Solid-state transistors and quantum transistors, bit and quantum bits, are usually considered to be very promising in terms of scalability. You can make these devices very small, compact. You can scale them up all using well-established nanotechnology tools. And there are different candidates for solid-state qubits, starting from electron spins in semiconductors, superconducting systems, nuclear spins, very different systems that share this advantage of being scalable and controllable in a fast time scale. At the same time, this system usually suffer from inhomogeneous environment, especially for quantum bits. This is usually a challenge, and typically also show relatively short coherence time, often to a very complex environmental interaction in solids. Today, I would like to show you potential of one particular solid-state system that is combining advantage of atom-like qubits and ability to place this qubit in a solid-state environment, having a long coherence time and very robust optical readout. The system I'm going to talk about is color center in diamond. Today I'm going to focus on one particular color center, nitrogen, at the carbon lattice location and vacancy nearby. This dopant in diamond creates kind of a molecule inside of diamond lattice, and this molecule artificial molecule can absorb and emit light when present in a high concentration and give this color to diamond crystal. And we are going to use this absorption and emission of light for visualization of color centers and reading out internal states of these atoms inside of diamond lattice. Given a few more details about these optical transitions of color centers, I have to say that this nitrogen vacancy color center is particular because optical transitions connect ground and excited state which are spin triplet. So essentially optical paths connect long-living spin sublevels, and this is what is essential for making diamond spin qubits. So it's a spin qubit in the ground state of this color center, now being readable and initializable using optical transition. And so how you basically use this combination of optics and microwave, which are combined now in a single color center, well, you basically shine the light to excite this color center, to promote the electron from the ground to the excited state, and owing to some internal dynamics, one of the spin states shelves to a singlet state of this color center, which is a dark state. And this paths lead to emission of less photons compared to another spin state with magnetic quantum number 0, this ms=0 state, which emits more photons. So now if you shine a laser pulse and measure fluorescence intensity versus time, so depending on which state you start, you will have more fluorescence, like in this red curve, or less fluorescence. After some times, you will reinitialize the spin state, but this initial dynamics is carrying information about the spin state. So you can essentially get access to information about the spin state of the color center by measuring fluorescence. And this technique is quite sensitive. You can repeat these measurements many, many times. Typically, for this optical readout, we do need to repeat experiments many times because we don't collect all the photons. But this technique can be so sensitive that after several repetitions, we can detect spin states even of a single color center. How this experiment looks like, I can show you a scheme. So we typically place our color centers in an ordered way, depending on application, and we approach high collection efficiency optics. We excite very small volume in diamond, having just a few color centers, and collect fluorescence light using efficient avalanche photodiodes. To control the spin state, we can place microwave line that excites spin transitions. And now if you plot fluorescence intensity of one of these spots when sweeping microwave, you will have a dark resonance. This is exactly the situation where you pump the population from the bright state to the dark state, and that's lead to lower fluorescence intensity. So measuring brightness of the spots, single atoms in diamond, single dopants in diamond, you can measure magnetic resonance on a single spin state. And that's quite a significant improvement of sensitivity compared to conventional spin detection. So you really can approach the level or can reach the level of detecting single dopants, and that's mean detecting single spins. So this is a photo of the experiment. Typically, we use synthetic diamonds with these small microwave structures on top of this. The area where NV centers are implanted is somewhere here between these microwave lines. We can also control the spin state coherently by applying short microwave pulses and excite Rabi oscillation starting from eigenstate of this spin system going to another eigenstate, but we can also stop the evolution at particular time, exciting a coherent state of two spins. And again, measurements here are performed optically. So we measure fluorescence intensity, but these two levels of intensity, dark and bright, correspond to two different spin states of the single color center. So this is a toolkit we use to manipulate the inner state of single atoms, and ability to control individual quantum system and read them out nowadays attract attention in context of application in quantum information science, in quantum metrology, in quantum simulations. And today's presentation will be about use of diamond spin qubits, use of color centers in diamond for making elements of quantum computer and making very small and sensitive sensors for magnetic field. I will start with quantum information processing and will show you design of diamond quantum processor. So this is about coupling spins. You have seen that we can read out spins very well, but now in quantum computer, you basically need to couple many of these quantum systems in a controlled way. And our design is array of NV centers in diamond. So each NV center couples to next one via magnetic dipole-dipole interaction. And in addition, every NV center might have some kind of a quantum memory, a long-living memory to enhance the functionality of this quantum processor. So today I'm not going to talk about this memory elements, which is interesting field on itself, but I'm going to talk about how to couple electron spins of neighboring atoms via magnetic dipole-dipole coupling. So, and if you want to couple qubits, you need to place them close enough that interaction between them is stronger than decoherence. So interaction between NV centers should exceed decoherence rate. And in realistic conditions, in our scenario where we have some environmental interaction, we need to place these color centers as close as a few tens of nanometers, maximum about 50 nanometers. And so placing these color centers in this range and addressing them with optical techniques is currently in challenge. So this is again to illustrate this distance challenge. So here I plot the interaction strengths between two spins in hertz versus distance, two single electron spins. And you see that if you want to have your quantum processor to work at least kilohertz rate, you need to be as close as a few tens of nanometers. So this is now our goal, to place these color centers very close, explore the interaction, read these color centers, and do it in a very well-controlled way to reach high fidelities. And so this interaction need to be stronger than decoherence rate. So need to be need to be more precise than a few tens of nanometers. There are technologies how to create these color centers on demand. Most of them are related to implantation of atoms inside of diamond. So you take very pure diamond and you basically focus ion beam and do implantation one ion after another, either by focusing the beam or by implanting atoms through small aperture. We can also do it on a large scale, having kind of nanolithography written structures on top of diamond. And current state of the art is about 20 nanometers accuracy. So one experiment we have performed in collaboration with a group of Jan Meyer in Leipzig was actually using this nano aperture technique The small holes were created in a mica placed on top of diamond. So here you see electron microscopy picture, and nitrogen ions were implanted through this very thin, very narrow holes. And here you see array of implanted color centers. We were targeting to have 2 color centers per spot. And we have post-selected the right spots afterwards that have 2 color centers that are placed close enough to start to feel the interaction. And here I will show you now a spectrum similar to that you have seen before, but now from 2 color centers. So this is a spectrum on a right down corner of the picture. You see several lines. So spectrum is complex because you have now 2 color centers. So the color center A is responsible for outer line. Color center B is for inner line. So this splitting is related to nuclear spin of N15 nuclei inside of NV center. And actually magnetic coupling between color centers would be another splitting in the spectrum, but it's not visible in this picture. In this case, 2 color centers were placed about 20 nanometer apart. Here you see a super resolution image, and that corresponds to a few tens of kilohertz coupling. So we can see this coupling now, and I will show you experiments how to visualize this. Basically, we use one NV center to measure the magnetic field created by another NV center. And so by switching state of one of the NV centers, we can see the shift of the resonance line of the second color center. So this 3 line position correspond to 3 eigenstate of one of the NV center, now measured by the second one. And this is a key element to create 2 qubit gates, to create conditional dynamic of one spin depending on the state of the other. So this is very basic element for scaling up quantum processor from 1 qubit to many qubits. So currently, the precision in positioning color centers is still a challenge, and we are working on improving it. I will show you a few recent efforts in this direction. So one of them is to use basically better focusing of ion beam, and to better focusing requires some novel technology in creation of very well precise beam of nitrogen ions. So here we collaborate with team of Jan Meyer, expert in implantation of nitrogen ion, and team of Ferdinand Schmidt-Kahler and Mainz, expert in controlling ions in the ion trap, also for quantum computing applications. And so we have proposed and recently realized a novel ion implanter where the source of ion is a Paul trap. The Paul trap that is used for making quantum computers out of ions, but now it's used to make a deterministic source of nitrogen. And so we basically trap nitrogen ions inside of this trap and then release it. And because of very cold temperature of nitrogen ions that we can achieve, we can create a very well-defined spot during implantation. So I show you a short movie how this works. The trap which is shown here will be loaded by the, so let me see, so I can start the movie. Yes. So the trap will be now loaded by ions, and one of them is calcium. Calcium is used to cool the system, and next ion is nitrogen. And after laser cooling, we basically release first calcium ions, leaving nitrogen, and nitrogen now very cold, it's sympathetically cooled, and this is now released and implanted in diamond lattice one by one. And this positioning can be very precise, down to single-digit nanometer, owing to this very well-defined wave function of the ion in the trap. And here you see experimental results. Picture showing the NV centers now created by these implantation techniques. Actually, we were targeting to have this nice array, but you see some disordered spots. Each of these spots is actually, we can confirm, is coming from ion trap, because we implant rare isotope of nitrogen and 15, but many missing spots, basically not making possible to see the array, are related to low yield of creation of NV centers. So at the moment we can very well implant nitrogen in diamond, so with 100% accuracy, but not every nitrogen is forming in NV center, and this is currently a challenge in this field. I would like to highlight another way how to create very well defined clusters of NV centers with even smaller distances, and means a better coupling. This is a technique that was pursued in collaboration with Takeshi Oshima and Shinobu Onoda from QSD in Japan, and Professor Tiraji from NIMS in Japan. And in these experiments, we were implanting not single nitrogen ions, but molecules containing many ions. So this adenine molecule, when being implanted in diamond lattice, create 4 nitrogen dopants, potentially up to 4 NV centers. And the distance between these color centers can be very small, because a molecule is a single point entry source. Of course, there is a struggle after you plant a molecule in diamond lattice, and ions are getting a bit separated, but so initial, this entry point is ideal point-like source. And so we have performed this implantation with different types of molecule. I will show you example for adenine having 4 nitrogen in the structure. And so what you see here is again the spectrum of this NV center cluster, in this case pair, created by adenine implantation out of 4 nitrogen, 2 formed NV centers. And so we can actually now benchmark coherence time and the coupling between these NV centers. On the lower graph, I show you oscillations of this basically pair internal state related to coupling, given about 30 kilohertz coupling strengths. On the middle graph, I show decay of Han echo, spin echo, which show coherence time on the order of a couple of 100 microseconds. And this ratio, so the time you need for gate, which is inverse coupling strengths, and coherence time defines how a quantum is your pair, how good this pair can be used for quantum computing purpose. And so in this case, this ratio, gate time, which is about 15 microseconds, versus coherence time, which is beyond 100 microseconds, is already very promising, especially in context of solid state system. Here I show you again this quantumness of this pair. I show decoherence rate on y-axis versus coupling strengths, and basically coupling parameters which are above this line is more or less incoherent coupling. So decoherence is higher than stronger than the coupling between spins. So the good spins that are coupled stronger than environmental noise affect them is somewhere lower. And I put here some set of reported data, and this pair that you see here is somewhere here, the red circle. So it's basically significantly beyond this line, and this coherent coupling can be now used for creating quantum correlations and performing small quantum algorithms. Another point, basically, of course, go to larger systems. And using the same technique, we also have been able to create triple NV centers. So we have now 3 qubits in a system. It's now a more complex spectrum. We still can address all these spins in a microwave domain. And currently, we are working basically to extend this number from 3 to 4 to 5 and scale up these quantum registers to larger number of spins. So low yield is still a challenge. And there is a strong material science effort in improving this yield. For example, understanding the charge state of defects and their influence on associations between vacancies and nitrogen. So basically controlling the charge state of dopants and using this control for formation, more efficient formation of NV centers is a current effort of the community in this field. Now I would like to tell a few words about readout. Remember, we need to control these spins, and you have seen how we can do it, but we also need to read out spins in a very efficient manner, sometimes also in nanoscale. And so far, optical technique was used as a workhorse. Optical technique in diamond have some drawbacks because most of the photons are not leaving diamond. If you excite in this center and it sits below the diamond surface, a lot of emitted photons are totally reflected on the surface. Only a small fraction leaves the diamond. And there was effort actually how to improve this by curving lenses in diamond as one of the possibilities, making cavities to improve the emission and make emission more directional, like this nanowires or integrated one-dimensional resonators. We can also improve actually the emission rate using cavity. And this is, I think, very promising avenues, how to collect photons more and more efficiently. But today I would like to talk to you about another approach, namely a hybrid approach to read out spins based on a photoelectrical effect. And this is collaboration with a group of Milos Nesladek at Hasselt University, Michael Trubke at Technical University of Vienna, Adam Gali at Wigner Institute in Budapest, and Takayuki Teraji and Junichi Zoya at NIMS and Tsukuba University. So the idea to improve the optical readout is related to photoionization dynamics of these dopants. So when I was talking about energy levels of color center and diamond, I was showing you before this scheme, but both energy levels, ground and excited states, in a solid diamond are located between valence band and conduction band. And so when you excite the color center, if you do it fast enough, you can also promote the electron not only to the excited state, but also take the electron from the excited state and promote it in a conduction band. Now, diamond being insulator at this moment become conductive. So you get photoelectron in a conduction band. And with this, you lose your NV center, you kind of ionize it, but then the electron can be taken from the valence band and recharge your color center back to this kind of original negatively charged state. During this process, you absorb 4 optical photons. So you need 2 to photoionize and 2 to recharge. And you get 1 electron and 1 hole in diamond. And this makes diamond conductive. So now if you apply voltage, you can measure photoelectrons and photoinduced holes. And so this is basically the idea behind the readout, which is not optical, but how it's called photoelectrical. So it's electrical assisted by optical excitation or photoionization. And this can be now very efficient because you can put very small electrodes very close to your NV center you might basically be able to collect photoelectrons and holes more efficiently than you collect photons. And here I show you the illustration of this. On the left side, you see the image of color centers. They are located between electrodes. Electrode is on the top and down part. And so in between, you have these spots. Each of them corresponds to emission of the single color center. And on the right side, I'll show you the picture of photocurrent when you raster the laser spot. And you see one-to-one correspondence between these two pictures. So the single color center visible here can be also visualized here. And we basically, if you compare the quality of the image or the noise or the number of detected charge carriers compared to photons, you can see that actually photoelectrical readout can be superior to optical readout. I zoom in here to a single NV center optical picture or fluorescence picture on the top and compare it with the photoelectrical picture on the bottom. And if I plot here number of photocounts, the top versus photoelectrons, so there is a significant improvement in the signal because you can collect your photons less efficiently than you can collect photoelectrons. This is one reason. Second one is that number of photoelectrons that you can produce this way is not necessarily limited by decay of the excited state. So here I give you a little bit more insight of this. So here I show the intensity of the signal of the single atom and put it as a function of the laser power for fluorescence on the right side. And this is the spin curve. And for photoelectrical readout, this is this curve, rising here, reaching tens of millions of photoelectrons per second. And what you see is that optical signal saturates at high laser power. And this is just because a two-level system like an atom cannot give you more photons than inverse excited state lifetime. So when you reach this value, increasing laser power further on don't help you to get more signal. But photoionization is stimulated process, is not spontaneous emission. So this rate can rise beyond the inverse kind of decay of some state related to spontaneous decay. So there is also a difference here, and you can get signals to be stronger. In addition, so photoelectrical readout rely on ability to collect photoelectrons using electrodes, and those electrodes can be placed very, very close to color center, closer than diffraction limit of light. So with this, I will switch a little bit the gear, and in the second part of my presentation, we'll show you the area where the even single color centers, not many of them which are needed for quantum computer, but even single one can provide you a regime where you reach quantum advantage. And my example that I'm going to bring here will be the use of single spin associated with color centers for magnetometry at nanoscale. So we have this very nice control of single NV centers, and we have access to its spin state. So the spin state is sensitive to magnetic field, and you can use it as a kind of compass now. If you approach it very close to other spins, you can visualize them, you can study its properties, and measuring a magnetic response of materials at nanoscale is carrying very important information about materials with many, many applications. I'm going to talk today about applications in nuclear magnetic resonance. But first, maybe just a few words about making sensors out of qubits. If you look on the energy levels of these color centers, you have seen spectra already before. I just now put emphasis on evolution of energy level of spin state as a function of the magnetic field. So if you rise the magnetic field, the energy level split, and the splitting is actually proportional to the magnetic field. So if you know the G factor or magnetic moment of your spin, you can calculate the B field by measuring this splitting associated with Zeeman effect. And this is a very general basic of construction of atomic magnetometers for a variety of systems. So we again measure our optically detected magnetic resonance, now track the spin transitions. Now we are not using them to access spin qubits for quantum computer, but we use the frequency information about the splitting to access the information about the magnetic field. So how precisely you can do this, how precisely you can measure the magnetic field depend on how precisely you can track the frequency of the spin transition. If you look into this in more details, you can find out that there is a fundamental limit associated with line width, or in other words, with coherence time. So if you use a quantum system like a spin and you want to measure magnetic field, you basically, in the most efficient way, try to excite coherence between two spin states and then measure phase associated with magnetic field. This is the equivalent of this line shift, and the smallest possible magnetic field that you can measure, which is equivalent to a noise of the spin by measurement, is related to magnetic parameters of the atom, of the spin, the number of atoms under square root here, measurement time tau, and coherence time big T. So if you have longer coherence time, you can improve the sensitivity. If you have more atoms, because they act kind of as amplifier of the signal, you also can reach better sensitivity. In many experiments where we are trying to target nanoscale, we use just single atom, and then the sensitivity is solely limited by coherence time. It's called the standard quantum limit. And so what about NV centers in diamond? How good is this coherence time? So here I show you the example of the Ramsey fringes decay. It's experiments where you excite coherence and then you leave your spin to process and you measure the state and the phase that it was accumulated. It's basically now information about magnetic field. And so here you see this decay of this oscillation of precession, and this decay is visible basically on a few hundredths of microsecond coherence time. So it's now the best point to measure magnetic field. It sits somewhere here and measure the shift of the fringes. Now, what does it mean in terms of magnetic field in Tesla or nanotesla or microtesla? So if you take these atomic parameters, so you can reach actually with a single atom nanotesla sensitivity for magnetic field. And how good it is compared to other magnetometers or quantum magnetometers. So here I show not only quantum, but all the magnetometry devices available in terms of two benchmarks, sensitivity as y-axis and effective dimension of them, which give you also limit to ability to approach them very close to the object of interest. And so you see that diamonds are holding records somewhere on a very small size scale. So reaching nanotesla here. They can be also interesting for larger devices. For larger diamonds with many NV centers, sub-picotesla sensitivity was reached, but today I'm going to talk about experiments at nanoscale. In a regime where it's very important to approach your sensor to the object of interest, in our case, it will be nuclear spins of molecules. So the experiments I would like to introduce right now is about measuring AC magnetic fields associated with precession of nuclear spins, kind of MRI settings, but now at nanoscale with very high sensitivity. And so in order to do this, I introduce one toolkit. I need to introduce the experiment afterwards. It's how to make a spectrum analyzer out of this magnetometer. So we were discussing before that essentially we are tracking the shift of the line, or we are tracking the phase accumulation after introducing or creating coherent state. So if you want to now make your sensor sensitive to only one particular frequency, you introduce many pi pulses that refocus evolution of your sensor. And you can construct this sequence in a way that your sensor will only be sensitive to one particular frequency of magnetic field. So you create a spectrum analyzer. So similar to optical spectrometer, where by placing a grating, you create a positive interference for certain wavelengths, a certain frequency of light to a given direction. So here, this is done in time domain by placing these pi pulses. Only one frequency will not be refocused by this train of pi pulses, and it will be imprinted as a phase on your spin that is used as a sensor. Now, the experiment that we were targeting as a first demonstration of this magnetometer is a demonstration of sensing of external nuclear spins placed on diamond. In this case, we were using oil, heavy hydrogen, and hydrogen is also carrying spin, but nuclear spin, so much weaker than actually in this center electron spins. But like in the case of quantum computers, these spins are starting now to interact, and these hydrogen nuclear spins are creating AC fields at the location of NV center. And so by creating this sequence of pulses, we can make a spectrum analyzer, and by changing time between pulses, we can shift the sensitivity function through the spectrum of the Larmor precession, and we can detect this Larmor precession of a very, very small volume of nuclear spin. So this sensitivity of NV center to nuclear spins can be reaching a level of single nuclear spins. And this is not because NV center is the most sensitive magnetometer, it's because you can bring it very, very close to the nuclei. Actually, even from a few nuclear spin at a distance of 1 to 3 nanometers, the magnetic field associated with Larmor precession can reach microtesla. So this is a very interesting regime where nuclear magnetic resonance can be now recorded using a quantum sensor like NV center and diamond. And contrary to conventional NMR or MRI, it's now a nanoscale. And this is possible because of different sensing regime. So you sense magnetic fields with very, very small sensor instead of sensing the induction using a large coil. What are the applications of this? Actually, measuring NMR give you access to a very interesting structure and chemical parameters of the system. This is particularly interesting in context of biosensing. Think about having biomolecules attached to diamond, and having this NMR allow you to measure the properties of biomolecules, for example, of proteins. And one demonstration that was already realized was done in collaboration with Mikhail Lukin group at Harvard. And in this case, the stable ubiquitin protein was attached to diamond chemically within V center, now implanted just below the surface, a few nanometers. And this chemical attachment was done by functionalization of diamond surface with specific groups. You start with carboxy, and then you change to amino group, and then protein has some complementary group that can chemically bind to this. And here you see atomic force microscopy pictures of diamond before this chemical binding and after this chemical binding. These bulbs here are single proteins. They're very dense, roughly monolayer, but a single NV center sit below one of them and sends a signal out of a single protein. And here again, these spectrograms. This is essentially magnetic field of y-axis and frequency on x-axis. It's inverted frequency. It's a time between pulses. And these peaks here correspond to now nuclear spin associated with structure, composition of these protein molecules. And this is a single protein molecules. In a conventional MRI or NMR experiments, you would need billions and billions of copies of this. And now this very small sensor allow you to reach ultimate sensitivity in this experiment. So this is detection, but learning about the molecule itself, for example, its structure, it's still a challenge in the field. If you look at a conventional NMR experiment that is used to elucidate the structure of the protein, it actually uses very high spectral resolution. Each of the spectral lines here carry information about particular spin. And so understanding the splitting allow you to reconstruct the structure. And it's not only about reaching a very high sensitivity, which is important to capture structure very quickly, because protein can also have some dynamics, but it's also about reaching high spectral resolution. And in the previous experiments that we have seen, the spectral resolution of this sensitivity function was given by coherence time of NV center itself. It can be, you know, like a millisecond maybe, but it's give you coherence time or limited by coherence time resolution of a kilohertz. And we are looking to resolve spectral features of a few hertz of resolution, maybe a few tens of hertz. And so this is basically the main avenue in this advancing this field is to improve this parameter, improve the spectral resolution and improve sensitivity to make spectroscopy faster. There are some very nice examples how chemical composition of molecules can be already reached within this technique. And I show here the work by Stuttgart group, led by your Prague group, where fluor nuclear spin were detected in a very small volume, again, nanoscale, and you can detect different fluor atoms with this spectroscopy, now getting information about the chemical structure of this molecule. And that was done by detecting a chemical shift. There are more shifts in NMR spectra. So chemical shift is the largest one that was detected, and it can be used to understand primary and secondary structure of proteins. More details about structure of proteins can be unraveled when measuring J coupling, and this is about a few hertz to a few tens of hertz. And basically, we need tools basically to go to this area where we can measure signals with spectral sensitivity down to hertz or even better. And one of the ways to do it is actually improve the protocols. I won't give you so much details about this, but just want to tell you that with better pulsing, with better pulse sequences and clever placing pulses, mixing the data acquisition with a timestamp from local oscillator, you can make your signal to be dependent not only on the frequency of the signal, but also on the phase of the incoming AC signal. And if you make your detection to be phase sensitive, you can beat this versus local oscillator phase. And in this case, your spectral resolution can be limited not by the coherence time of your sensor. So we're limited to this region, so 1 hour this time, but to the stability of the classical local oscillator, which can be much better than this. And I show you here example where 3 spectral components were resolved with a resolution of millihertz, and so the components were separated by a few tens of hertz, and they're very nicely resolved using this NV center sensors. Saying this, and maybe giving a little bit of feeling of future applications, I would like to highlight that nuclear magnetic resonance is not the only signal. So if you can measure magnetic fields at nanoscale, you can think about understanding the flow of ions from the membrane. You can also measure electron spins in biomolecules, and those are often related to catalysis. Or you can attempt to measure magnetic fields associated with propagation of action potential in neurons. In all these cases, the magnetic field that is associated with these processes is within the reach of sensitivity of NV center quantum sensor, quantum magnetometer. In many cases, you would like to really look inside of cells, and there can be 2 ways to do it. Current experiments are done with cells or basically placed on a flat diamond with NV centers located very, very close. And many of this demonstration of nanoscale NMR are done in this setting. But you can also think about going inside of cells, because NV center experiments don't require any vacuum, and you don't need to cool your system to low temperature. So you can think about taking a diamond particle, functionalize it to target certain proteins, a certain area in cell, and then insert it inside of cell and turn this whole experiment inside of a physiologically relevant intracellular environment. And so the crucial point here is how to create these nanodiamonds and keep NV centers inside alive and not deteriorate coherence time. And this is basically a current effort. There are different types of techniques that allow to create nanodiamonds, either by detonation or by milling diamonds. And it's also very important to control the surface of diamond and functionalize it, either by chemical groups directly linked to the surface or by creation coating non-covalently bound to diamond and based on a kind of a polymer. And then basically these diamonds can be transferred inside of cells, either by injection or by endocytosis, where a cell takes the particle. And here I show you experiment inside of cell with this antenna, but now not big diamond, but a cell located close by. And these bright dots are now individual NV centers, individual qubits, but now used as a sensor inside of cells. So you can do very similar experiments we have seen before. You can create radio oscillations. You can see decay of echoes related to environment inside of cells, and this environment carry information leading to relaxation between spin sublevels, carry information about chemical processes inside of cells. So with this, I would like to conclude, maybe again highlight what you have seen, applications of color centers for quantum computer, and a few qubits are reachable already with pretty precise gates, which is also essential. And basically on the material science side and the hardware side of this device, we still need to create these qubits with more precision and more yield, better yield. I also would like to highlight that also basically microwave pulses and control of the spins need to be optimized, and optimum control tools are very essential for both protecting your qubit from environmental noise, but also for creating very high fidelity gates. And this both hardware and software improvement will be very essential for going to next level of fidelities of gates in this quantum register. Here in the arrays of spins, we would need many qubits to reach quantum advantage. But in a field of sensing, even single or a few spins already bring you to a regime where you reach quantum advantage for a very small qubit system. And so I think this is where the first applications might come. We have discussed life sciences and nuclear magnetic resonance. Actually, the spins can be also used to enhance MRI by transferring polarization from NV center to external spins. But life sciences and medicine is not the only one. You need sensitive magnetometers in a variety of fields, from space research to navigation to communication, making better radars. All this is within a reach of interest from other communities to this sensing. This I would like to thank you for your attention, and hope we have a bit of time for questions. Thank you.
Thank you very much for this nice overview of both experiments and application of technological application in the quantum world. So now I should say that if somebody wants to ask questions, there are two ways. You can either raise your hand or use the raise your hand button, or the Q&A or domande risposte to raise your question, then which can be then sent to the speaker. So please. Let me see if I see some question. I just have a curiosity. You mentioned the space research. What do you mean by this space research in its application?
So basically, good magnetometers are very essential for understanding planets. So even, in the first Mars zone, there was already a magnetometer. So sensitive magnetometers that are not requiring a lot of energy, not cryogenic, and having been calibrated, self-calibrated, it's very essential. And we do have collaboration with German Space Agency to make these small diamond magnetometers, not as small, not nanoscale, but more millimeter size for space missions. That can be important for other planets or moons, or it can be important actually even for exploration of the magnetic field of the Earth on our orbit. So both are important questions.
So maybe here I have a follow-up question. So in that, first of all, thank you very much for your nice talk. And so there, the important is to have small diamonds so that you put them in orbit, or also this enhanced sensitivity.
I think both parameters are important, to be small, to be on orbit, and to have a high sensitivity. I think at the moment, the size of diamond is still not very crucial, because the periphery, like a microwave source and this laser source, are still larger than a diamond. But it can be basically integrated in a size of a few centimeters, all these experiments. And that's already get basically an interesting regime for this. Then, of course, for space is kind of tricky because it needs to withstand the launch. So the acceleration and vibrations, electronics, diamond is very resistant to radiation, but the electronics need to be certified for this, and there are a lot of other things that comes along. So it takes very long time to develop something for space, but in general, so if you put the numbers and sensitivity and size and take this self-calibration as a bonus, so this is definitely within interesting device set that can be sent to orbit or beyond orbit.
Good. So, and maybe I have another question if there are no others. I don't see. Chair allows me. So, in the first part of the talk, you were showing us how you are building up your quantum register and the nice developments in the number of MD center that you can create and control and couple. So, do you think that are there fundamental limitations there? Or with a due development of a technology, we will be able to really scale even this platform up to compete with the other quantum computing platforms?
So this is a very important question, whether the limitations are fundamental or of technical character. So currently, it's actually the yield of creation of color center is a limitation. So when we implant, so we just have a conversion of a few percent typically, and it is a limitation. Now, it's a technical or fundamental. So we know that it is possible to have also 2 or 3 color centers that are coupled. But improving this yield, it is of kind of a technical character, but it's an important material science question. So how to add more vacancies, to have enough vacancies to associate with NV center. So it's basically providing more vacancies in diamond lattice in the right point, right time point, and also providing enough charges, because this NV center need to be negatively charged. So we need another donor somewhere. Also basically bringing these donors in a diamond lattice in a right concentration. So this is another another point that need to be addressed. And so I think it's in principle technical point. So it's basically engineering diamond lattice, but so it's very important to address them. And it requires systematic studies of diamond material.
Yeah, good. So eager to see how this will develop.
So So I just another small curiosity. Do you think that you have application also to neuroscience or?
Yeah, so there are applications to neuroscience. There are two sides of these applications. One is to take a piece of diamond and place a single neuron on top, or maybe a tissue. But so in vCenter will be in close touch with a very first cell. And in this case, you kind of now, when you have a propagation of potential through the cell, so you can estimate the magnetic field. So because the current is kind of flowing, there is a magnetic field, and near the membrane, it can be up to a fraction of nanotesla, according to model. So nobody measured it this way. Typically, people measure electric fields, but not the magnetic one, and it's complementary information. So that could be one experiment that is of very big importance. The second is basically not looking into the single cell, but having this magnetometer emitted part, but now from brain, not really of a single cell, but approach experiment more medical relevance, replace QID in magnetoencephalography. So it's basically measuring the magnetic field of the actin brain instead of electric fields as a signal. And currently QID is the only technology that can do it. So now atomic vapor magnetometers are coming into this field with the first demonstrations, not yet commercial as far as I know. And now diamond can be another system that is not requiring, like shielding and so on. Maybe not as much shielding as other technologies, not cryogenic, so can be very mobile. And there are even visions basically in this application to have it as a helm and to read the neuronal signals that can be essential for a variety of applications, medical and non-medical.
Thank you very much.
There is one raised hand, Michele Merano. If you want, I can allow him to talk.
Please.
Yeah, I have just a curiosity. You showed this measurement with single molecules. I wonder how the magnetic field vary with distance from this is known from the.
Yes, absolutely. So it's very important question here. So it's maybe I'll share very quickly the screen, but there is not so much information about distance dependent on it, but at least just to give you the feeling about these dimensions. So it is kind of drawn in a dimension, the right dimension. So the vCenter is about a couple of nanometers below the surface, and the protein itself is a few nanometers. And so the distance between the protein and the vCenter is, well, depending on which side of protein, but it's between 2.5 and let's say 6 nanometers. The field of individual nuclei decays as a dipolar field, so the third power distance dependent. So that's why you don't see this protein on the right side, because it is producing the signal, but it's too weak. And so in vCenter, do see only close by nuclear spins. And so the signal itself is third power of the distance. It also tells you that the closer you get, basically, the faster you can measure. But it's sort of the settings. If you would be with a single in vCenter, if you would be a micron apart, you won't have enough field to measure. So that's too weak then.
Other questions? I don't see another questions. So I think we thank very much you for this nice talk, and I really hope to see you in Padua in next time.
Thank you very much. It was a pleasure for me, and looking forward to visit your department.
Thank you.